Yeah. Here is the topic: development of Gibbatrianth mycoherbicide to control Trianthema portulacastrum, horse porcelain, and notorious agriculture weed. When we talk of this horse porcelain, it is the agriculture weed, not of the our country, India, northern part of our country. It is also an agriculture weed of Pakistan, Mexico, as well as the United States of America. Because the first discovery of this fungus Gibbatrianth, that is the Gibbatrianth, was made on this very weed from the United States of America itself in 1986. Go ahead to the second slide, second second slide, please. Next slide, next slide. Go to the next slide, please. Start viewing the slides. This is my I'm for a professor from Kurukshetra University. I'm alumni of this university. I started my that is the all qualifications, graduation, post graduation, and PhD from the Kurukshetra University itself. Joined in the teaching faculty and retired from the teaching faculty itself from Kurukshetra University itself. Next slide, please. Next slide. Next slide. Here is what are pesticides. Of course, all of us are familiar with the pesticides, what the pesticides are, and what the pesticides are doing to us, to the environment, as well as to the microbiota, and how these pesticides are going into our crop foods and they are affecting our health. Next slide, please. Next. This is this. This is the chain how when we are spraying these herbicides or pesticides in any of the ecosystem or agriculture ecosystem, how they go in our food and how they impact the flora, fauna, as well as the human health, and how they become the carcinogenic one. Next slide, please. Next slide. Next slide. This is, I'm talking of the pesticides, not of the pesticide, biopesticide, also called biological pesticide, which are the pesticide based on the microorganism or natural products. When we talk of these pesticides based on the target pest, these are classified as bioherbicides for controlling the weed, bioinsecticide for controlling the insect, biofungicide for controlling the plant pathogens or diseases, and bionematicides for controlling the plant parasitic nematodes. Next slide, please. Next slide. Next slide. Go to the next slide. Go to the next. These are the defecate. What the bioherbicides are? These are the hosts based on the whole host specific plant pathogenic microorganism, fungi, bacteria, and viruses for controlling the herbs or weeds. The way as we go for the spraying of the chemical herbicides are concerned. When we start with the discovery of the first bioherbicide, it was the Divine, which was discovered in 1981, and it was based on the fungus Phytophthora palmivora, and it was discovered from the United States of America. This is how the history of bioherbicides started in the world. And when we talk of these bioherbicides or mycoherbicides, bioherbicides are in formulation based on the microbes, that is the fungi, bacteria, and viruses, and mycoherbicides are based on the fungi alone. When we talk of the bioherbicides, the two terms are being used interchangeably because most of the formulations are based on the fungi. That's why we are using bioherbicide versus mycoherbicide. Go to the next slide, please. Next slide. This is when we talk why we are going for the bioherbicides. As you know, how the pesticides are impacting to us as well as to the environment. To have the safe, effective products are concerned, that is, they are safe, environmental friendly, prevent hazards, product the level of dependence, that is the on the chemical herbicides, and producing healthier foods and promoting natural farming. Next slide, please. Next slide. Next slide. Next slide. Now we talk of the global bioherbicide market is concerned. Lot of emphasis is there on the production of the bioherbicide in the world. As you know, the market of the bioherbicides in the world is $3 billion in 2024, and that is increasing at the rate of 10.86% and going to reach $8 billion by 2033. Next slide, please. Next slide. Here is, I'm talking of the total bioherbicides in the world is concerned. They are 26 in number. and most of them have been discovered from the United States of America, 10, followed by Canada, 6, South Africa, 2, and when we talk of the Netherlands, Japan, and China, 1 each product is available in these 3 countries. And when we talk of this as the product is concerned, this has been developed by from our country by us, but not yet registered, and we are in the process of going to register this product with the help of the industrial collaboration. Next slide, please. Next slide. Next. Here is the that is the information which I have given 26 mycoherbicide, that is based on the review which I have written, that has been published in last year 20224. That is the information based on this very paper, and this is the abstract of that very review, which is of 35 pages. Next slide, please. Next slide. Next slide. Here is the impact. We know how the weeds, if we talk of the unwanted or undesirable plants which affect the, you can say, agriculture crops in various ways. And when we talk of the weeds, they constitute about 3%, that is 8,000 in number of the total 250,000 species of plants which have been reported on the earth. These are the losses, how they affect grain, global economic losses, Indian losses as static nuisance, and pollens as source of allergy and diseases. Next slide. Here is the development, how the development of this journey of the development of the mycoherbicide started in our country or in the world as a whole. Next slide, please. Next slide. Here is the wheat, that this is the most troublesome weed in agriculture words, as already discussed in different parts of the words. Next slide, please. Next slide. These are the losses caused by this. This is one of the troublesome weed in the northern part of our country, and it heavily infects various agriculture crops starting mustard, maize, sorghum, sugarcane, cotton, and so many, and which cause losses in the yields ranging from 50% to 89% when we do not go for treating these weeds in the field is concerned. Next slide, please. Next slide. Here is the, that is the infestation, that how much this infest in the different crops. As you are seeing here, maximum percentage of occurrence has been reported in maize, followed by onion, and that is the pigeon pea. Pigeon pea is 90%, 87.5% is the onion, and 86% is the maize. And this is the infestation, and this is the percent occurrence which has been reported by us and different parts, that is the Haryana, Punjab, UP, northern parts of our country. Next slide, please. Next slide. Here is the, these are the figures which I have taken from the day, that how much losses, that is, you see the infestation and the losses caused by this weed in different crops is concerned. Next slide, please. Next slide. Here is the, when we talk of this, our journey starts with the, that is the infestation, that is the how much losses a weed is causing to different crops. Then we go to the, that is the fungal pathogens is concerned, how much that is the what are the fungal pathogens which come on this weed. When we talk of the total number of fungal pathogens which have been reported around the globe, they are 15 in number, and most of these pathogens have been reported from our country. But the first pathogen which was reported, as you are saying here, is the, this is the Cercospora trianthemi, which was reported from our country by Chidavar in 1962. But when we talk of the biological control of this weed through the fungal pathogen is concerned, it is through the Gibbatrianth, which was reported from US, Cuba, and Venezuela by Siemens in 1986 is concerned. Then second report of the occurrence, as you see here, was reported by us from India itself. That is, that paper was published in 1998. Then it was followed the biological. Next slide, please. This is the list of the total fungal pathogens which have been reported. The fungal pathogens have been reported from United States of America, from India, Mexico, Pakistan. These are the four countries from where these fungal pathogens have been reported. And if you see the table, maximum number of species have been reported from India on this very weed is concerned. Next slide, please. Next slide. Here is if we have to start working with any of the biocontrol agent as a mycoherbicide is concerned, first we go to the surveys for searching the virulent fungal pathogens. Then we go for testing of the pathogenicity and proving of the cause postulate. Then we go for the identity of the highly virulent isolates, and we do not go for the pathogens which are not causing virulence in the nature, because they are not going to control later on when we go for the development of any mycoherbicide is concerned. Then we go for the technology of the mass production of the inoculum, which can be used as a spray for controlling its mycoherbicidal efficacy is concerned. Then we go for the study of the life cycle of this fungus, and then the optimum conditions for the infection, for the disease development, for the growth, and for the sporulation of this very pathogen is concerned. Next slide. Next, go to the next. These are the then we go for the trials, first in the laboratory in vitro condition, then we go for the in vivo condition, then we go for the host specificity and toxicity tests, and then we go for the formulation and application technology, and then we go for the biocontrol potential under greenhouse and field conditions is concerned. Next slide, please. This is the protocol for the development of any microbial product is concerned. Next slide. Next slide. Next slide. There is this pathogen, this Gibbatrianth. I already told you that this fungus was first of all isolated from the United States of America by an American mycologist, Seaman, botanist from the University of Massachusetts, United States of America. This fungus is very interesting. This is a monotypic fungus that this fungal pathogen is having only one species, that is the Trianthema. This is what is this? This is a pheodictoconidial. This fungus is dark colored, produce dark colored conidia, which having transverse and longitudinal septa, and these are known weak, which means they are different from the another conidium alternaria fungus, which is also having the similar type of having transverse and longitudinal septa, which is referred to as a muriform conidium. But what is the another important feature is this fungus is having a constriction at the basal point of the tube. These conidia are produced by the polyblastic conidogenous cells, and these conidia produce the germ tube, and the germ tube have the ability to produce another conidia too. This is the conidiophore giving rise to the conidia. This conidium is giving rise to the another conidia in change is concerned. And based on the sexual reproduction structure, this fungus is classified in the deuteromycota, but based on the perfect state, now deuteromycota is not recognized, and it has been all the fungi which were earlier considered in the deuteromycota stage, they have been classified in the ascomycota now. Next slide, please. Next slide. This is how the conidial ontogenesis, that is the mode of conidiogenesis in this take place, that how you are seeing here small conidiogenous cell is producing, it is giving rise to a unicellular cell, it is divided by a transverse septum tool cell, it is divided by transverse and longitudinal septa and becoming the muriform. This one conidium is produced, it is still attached with the conidiophore, giving rise to the another conidium. This is another conidium, this conidial chain is being produced. You are seeing what is the important thing is the growth of the conidiophore doesn't stop, it continue genetically and give rise to the another conidia gene. This is the mode of conidiogenesis which we have studied in our laboratory. Next slide, please. Next slide. These are the symptoms when we go. This is the leaf spot symptoms which are produced in the fields as well as in moist chamber. This is the moist chamber plate where we kept the infected washed. These are the leaves plant washed with 70% alcohol. Then they are put in the moist chambers, incubated at 25 degrees centigrade, and then we see the development of the symptoms on these leaves is concerned. These are the leaves in moist chambers, and these are the leaves or the plants which are sprayed in the fields is concerned. Next slide, please. Leaf spot disease. Next slide. This is, we talk of the this fungus is a hemibiotropic fungus, and hemibiotropic means it is partially biotropic and partially necrotropic, which is having the partially biotropic and partially necrotropic. That fungus is called the hemibiotropic fungus. And what is the advantage of the biotrophy is that it remains, that is the specific to its host, it doesn't go to another host. And the necrotropic phase help in the killing of the fruits, it shows the biotrophy. First penetration, then followed by the biotrophic growth, then go for the necrotrophic or killing of the host plant is concerned. These are the three steps which are involved in infection and death of the any weed is concerned or any host plant is concerned. Next line. What is the most important thing is this fungus, when it infects or penetrates the host, it goes for the production of a what we call as the peg-like structure is concerned that penetrate the host, and that referred to as the aprisorium. Aprisorium is there. A swollen structure is produced when wherever it touches with the epidermal cells, and the peg-like structure is produced, and that actually penetrate the host is concerned or epidermis is concerned. Next slide. This is the mechanism of the penetration, how it penetrates. The first, as already, that is the first phase, penetration of the host. Then it is followed by the biotrophic phase. It goes inside, multiply the tissue cells, and then finally biotrophic phase. After that, when it multiplies in the mesophyll cells, then it goes third phase comes. That is the necrotrophic phase. That is the killing of the host by the production of the toxin as well as enzymes is concerned. Next, go to the next one. Necrotrophic phase. This is the necrotrophic phase, and it is the only when the necrotrophic phase, then the symptoms are produced, and before necrotrophic phase, it doesn't produce the symptoms are concerned. Then after the production of the symptom, it results in the what we call as the death of the plants. Next slide, please. This is the growth we have studied. If we have to see, we have studied for the production of the inoculum, which is to be sprayed in the field. This is, we have seen the growth of this fungus on 10 different agar media to 3, at which that is the what is the best medium for its growth. That is the colonial growth, that is the mycelial growth, and for the production of the sporulating structures or conidia is concerned, because these are the conidia which are going to be used as the inoculum for spraying. Next slide, please. We have found Trianthema extract agar as the best medium, followed by the PDA as the second best medium for the production of the growth and the sporulation is concerned. Here is the, we have studied the environmental factors, because environmental factors come in the way when we have to go for using any of the microbial agents as mycoherbicide is concerned. So this we have studied the incubation temperatures for its growth, and what we have found, we have used 6 different temperature: 5 degrees centigrade, 15, 25, 35, 45, and 55 degrees centigrade. And of these, we have set 25 degrees centigrade as the best temperature for growth as well as for its sporulation is concerned. Next slide, please. Next slide. There is the germination. Then we have seen as the germination of the conidia is concerned, what we have seen again, 25 degrees centigrade is the best temperature for the germination of the conidia. Although germination takes place 15, 25, and 35 degrees centigrade, no germination at 5 degree, 15, 45, and 50 degree, but the best is at 25 degrees centigrade. Next slide, please. Here is the earlier to that humidity again, effect of humidity, earlier to that effect of humidity on the, this is C, effect of this is the D, effect of humidity on this. This is going for the earlier to that, earlier. This is the temperature you are seeing here. Here is the what is the effect of humidities on the growth of the fungus, because again humidity come in the way when we have to go for using any of the microbes as biocontrol agents or as a mycoherbicide is concerned. So different chemicals were used for testing the humidity, and what we have found that the pure water is having the maximum, produces maximum humidity. In pure water, maximum, you can say, growth of the fungus take place at when we use the pure water, and followed by it is in this is sodium hydrogen phosphate is concerned. It is showing all these, but the best is the pure water. It means when we have to go use for the preparation of the inoculum, we should always prepare the inoculum in water is concerned, not in any of the other one is concerned. Next slide, please. Here's the biocontrol efficacy. Then we have studied the biocontrol. We have used that inoculum. We have we have taken these earthen pots, and then we have separated the inoculum of the fungus containing mycelium as well as the conidia is concerned under different environmental conditions. Then we have seen that is the how much after 50 days, but how much plant is killed after 50 days, how much after 30 days under this is under different conditions is concerned. Next is 80%. Then we have seen effect of the surfactant on the biocontrol efficacy. That is the only under water suspension was there, and here is the conidal suspension. It has been prepared in the surfactant, and surfactant uses 0.2% tween 20 is concerned. Then we have tested that it increases these tween increases the efficiency. We have seen that shows the cover control up to 70%, 75%. But when we use tween as the surfactant, the control is more than 90% of the weed plant. Next slide. Next slide. Here is the effect of temperature on this incident of the disease. No, what we have seen is the again 25 degrees centigrade is the best temperature for the development of the disease when we go for the spraying of the this inoculum is concerned. Next slide, please. Here is the again most important parameter. Two important parameters are to be used when we have to use any organism. One is the toxicity, another is the host range studies, and we have tested the host range of these Gibbatrianth on different plants belonging to the weeds similar to the that belonging to the weeds which are similar to the what we call as the water hyacinth is concerned, and then the crop plants in which this weed commonly occurs. Then we have found all the other plants tested are immune to this very what we call Gibbatrianth is concerned. Mean it causes disease only on the Trianthema portulacastrum and none of the other studies. Other authors have also reported that it doesn't affect any of the other crop plant is concerned. It is a host specific one. This Gibbatrianth. Next slide. Next slide. Here is the this Gibbatrianth on the basis of C that this fungus is first, it shows the hemibiotropic type of life cycle. And hemibiotropic life cycle, as already discussed, it is having both the biotropic and necrotropic phase. Biotropic has the advantage that it is going to be going to the host specific one, and necrotropic causes the extensive tissue of the death. Both the factors help in what we call controlling this very weed is concerned. Next. Here, what is the conclusion is that this fungus, Gibbatrianth, has all the desirable features which makes it an ideal biocontrol agent to be developed as a mycoherbicide. As it is a virulent phytopathogen creating epiphytotics in nature, it is easily culturable on a what we call as the cheap medium, that is the Trianthema dextrose agar. It easily saborulate, producing conidia on TDA. It is very aggressive to the weed and killing the host within that is the entering the host within 18 to 72 hours and resulting the what we call death after 8 to 10 days. It is a host specific and it can be mass produced on a short medium. And the formulation of the fungus, as the name is concerned, I have named it Gibbatrianth based on the what we call as the fungus Gibbatrianth is concerned, and on the wheat which comes with this Trianthema. Trianthema is concerned, that's why we have named it Gibbatrianth. So Gibbatrianth is nothing, it is a formulation of the fungus, that is the Conidia mycelium plus the what we call as the twin surfactant is concerned, that is called the Gibbatrianth. Next slide, please. This is the biography, that is the bibliography or the papers which I have used for the preparation of this very presentation is concerned. Most of the papers are mine. This you see on this work I have worked, because this is going to the first, this formulation is going to the first mycoherbicidal or bioherbicidal which is going to be developed and commercialized in our country. Next slide, please. Next slide. These are the, of course, these are the all publications. Most of them are mine. Next slide, please. Next slide. Next slide. Next slide. Now here is, as I already told you, from the core of my heart, I sincerely wish to thank the American Magnus Group, organizers of the GPB 25 International Conference, for inviting me as an speaker and providing an opportunity to present my work in an emerging area of research by waiving registration fee both online and offline. And I had a wish to attend this conference in person, however, missed the wonderful opportunity of meeting this conference and meeting the global scientists due to my time bound other commitments, and I couldn't the attend in person the conference which was held in 20225 by the GP group. That is the I'm thankful to the organizer, again very thankful to the organizer for giving me the opportunity to present, attend and present my work, of course online, in this conference. Any queries are welcome. Next 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 slide, next slide, next slide please. I'm extremely thankful to the listeners for the patience for hearing my talk. Now, here is a message that it looks to be a very simple work, but the journey has taken a very long time for the isolation of the pathogen identification, and then going to the in vivo, in vitro test at the pathogenicity cause postulate is concerned, then going to the field trials under different environmental conditions is concerned, and it is a work of not only single type of interdisciplinary work requiring the participation of what we call as the taxonomist or weed scientist is concerned, plant pathologist is concerned, biotechnologist is concerned, but not only thus, industrial collaboration is a must if you have to develop any of the product into a what we call as the bioherbicidal or mycoherbicidal product is concerned. And most of all, in addition to that, what is required is participation of the farmers, that they should come forward for accepting the product, because sometimes so many bio products are available in the market and farmers are not opting for them, keeping in view whether they will work or not, whether the efficiency will be slow or high. So that is, if we have to develop any of the product as a bioherbicidal product or mycoherbicidal product is concerned, participation of all is desired. And I wish this product come and we are able to produce what we call as the organic, healthier foods which are free from the chemical fertilizer or chemical herbicides is concerned. Thank you.